In this video, we're going to cover Practical 3 on testing for biological molecules. Our learning objective is to be able to use qualitative reagents to test for carbohydrates, lipids, and proteins. As always, all of the timestamps will be in the description below, so feel free to skip to whatever part you need help with. In this video, we're going to cover PAG 9, testing for biological molecules. Our learning objective is to be able to use qualitative reagents to test for carbohydrates, lipids, and proteins. As always, all of the timestamps will be in the description below, so feel free to skip to whatever part you need help with. In this video, we're going to cover required practical 4, testing for biological molecules, and our learning objective is to be able to use qualitative reagents to test for carbohydrates, lipids, and proteins. As always, all of the timestamps will be in the description below, so feel free to skip to whatever part you need help with. This is our only definition for this video. Reducing sugar, a sugar that can act as a reducing agent and donate electrons, for example, glucose, lactose, or maltose. Before you start this practical, you should be confident in describing the structure and function of the 3 main types of biological molecules involved in this experiment. So that's carbohydrates, lipids, and proteins. And you should also be able to give examples of each one. In order to prepare our food samples for testing, we need to use a pestle and mortar, as shown on the left, to grind up a small sample of the food. Once we've done that, we're going to transfer it to a small beaker or conical flask and add some distilled water. Then we're going to stir to allow some of the food to dissolve in the water and use a filter funnel and filter paper to obtain as clear a solution as possible. And then once we've got our solution, we're going to put that in a test tube and we can begin testing our samples. To test for starch, we need our dissolved food sample and some iodine solution. So the first step is to put our food sample in our test tube and then add a few drops of iodine using a pipette. If starch is present, you'll see a colour change from brown to blue-black. When we're testing for reducing sugars, we need to use Benedict's reagent and also a water bath, so the reaction happens more quickly enough. So we're going to add an equal volume or an excess of Benedict's solution into our food sample, which is going to be in a test tube, and then place it in a hot water bath for a few minutes. If a reducing sugar is present, a brick red precipitate is formed. If it's absent, however, the solution will remain blue. When we're testing for proteins, we need to add a few drops of Bure's reagent to our food sample. Bure's reagent is sodium hydroxide and copper sulfate mixed together. If protein is present, you'll see a colour change from blue to purple. When testing for lipids, we add a few centimetres cubed of ethanol to our food sample and then add some distilled water also. Shake the mixture together, and if lipids are present, a white emulsion is formed on the surface of the mixture. In terms of safety, we need to make sure that we tie long hair back, and when we're using the Bunsen burner and the water bath, and also in terms of the chemicals, ethanol is very flammable when it comes into contact or near to an open flame, and the Biore reagent contains copper sulfate, which is poisonous, and sodium hydroxide, which is corrosive. So we need to make sure to wear goggles, wash our hands if it comes into contact with our skin, and also wipe away any spills on surfaces. These are 2 common mistakes that students make when doing this practical, so make sure you remember these 2 facts. First of all, use a high enough concentration of your reagents for a clear colour change. As with the Benedict's reagent and the Bure reagent, the colour change may be subtle and difficult to judge if the concentration of the reagent is too low. Second of all, make sure you crush your food really well and perhaps use a water bath in order to get a fast reaction and a clear colour change as well. This is our first worked example question for this practical. The sugar molecule glucose can be detected by a chemical test. Use words from the box to complete the sentences. So we've given the words below. The blank reagent is added to a tube containing a solution of glucose. The tube is heated and the colour changes to a blank colour. So if we look at the first space, first of all, it's talking about a specific named reagent. So that means it's either going to be Bure or Benedict's. And the Bure test is used for proteins, so we're going to go for Benedict's. And secondly, the colour change is going to be from blue to brick red. And the lilac in the box is associated with the test for proteins. For our second worked example question, make sure to pause the screen and give it a go, and then I'm going to go through it. So describe how you would test for the presence of protein within a food sample for 4 marks. So let's get started on that one. So first of all, we need to increase the surface area of our food sample. So if we're given, a piece of meat, for example, we need to crush that up, and we're going to use a pestle and mortar to do that. So crush with pestle and mortar. Secondly, we need to make a solution out of our food sample so we can add our extra reagent to it. So we'll add some distilled water, and we're going to dissolve our food sample as best as we can in that, and try and get as clear a solution as possible. Then, because we're testing for proteins, we need to think of the relevant reagent for that test, which is going to be Bure's reagent. Then finally, we need to indicate what a positive test would look like. So Bure's reagent is blue, and if protein is present, our solution is going to turn purple. By the end of this video, you should be able to recall all of the following biochemical tests and explain how you would design an experiment to test for each one. Now go and try the practical yourself in class, and also do some practice exam questions.